Now we are going to pay attention to the different axes with just a little bit more detail. One of the most important axes is the gut-brain axis. When assessing the gut-brain axis, we should pay attention to smell, the ability to swallow, the duration of eating, saliva, any gut noises, the gag reflex, and also the ability to contract the palate at the back of the mouth. So let's have a quick look at smell. Digestion starts with smell, and when we look into the most common neurodegenerative disease, Alzheimer's, along with Parkinson's, this can give you a telltale sign. The first thing they lose is their sense of smell, followed by constipation. So looking at smell is going to be critical when paying attention to the gut-brain axis. This neurodegenerative process takes place in the gut 10 to 20 years before tremors even begin to start emerging, which means that you can have a tremendous head start when looking, tremendous is a bad word to use here. This neurodegenerative process takes place in the gut 10 to 20 years before tremors emerge, which gives a humongous head start into looking after health, particularly that of the brain. When dendritic cells in the gut become inflamed, microglial cells in the brain also become inflamed, and there's this direct interplay from the gut to the brain. In fact, we're going to be looking at this much more, not only in this seminar, but in another seminar looking into brain health. In addition to the classic gut-brain axis, there are also others to be discussed. As we mentioned previously, there's the gut-skin axis, and this is often recognized through the clinical presentation of things such as psoriasis, atopic dermatitis, and even acne. Through complex immune mechanisms, the influence of the gut microbiome extends to involve distant organ systems, including the skin. With the intentional modulation of the microbiome, probiotics, prebiotics, and symbiotics have proven beneficial in the prevention and/or treatment of inflammatory skin diseases, including acne, dermatitis, and psoriasis. We also have the gut-liver axis, and we can see that there is this association between things such as liver injury, cirrhosis, or even liver carcinomas, and the microbial state within the digestive system. An accumulating body of research suggests that the disparate observations in liver disease-related studies are unified and explained by the microbiome. It is now widely accepted that liver damage can result from extensive interplay between the gut microbiota via specialized molecules such as TMA, acetaldehyde, and lipopolysaccharides, and the host immune system via Kupffer cell-mediated liver inflammation. So these Kupffer cells can switch on inflammation when they are exposed to certain molecules, and these molecules can be produced, such as acetaldehydes or LPS. They can be produced within the gut, which therefore exacerbates and switches on that liver inflammatory response. Now, there's this bidirectional communication between the gut and the liver, and this can be through the biliary tract and also the portal vein. The liver transports bile salts and antimicrobial molecules, such as immunoglobulin A and angiogenin, to the intestinal lumen through the biliary tract. This process maintains gut eubiosis by controlling unrestricted bacterial overgrowth. Bile salts also act as an important signaling molecule via nuclear receptors, such as FXR and G protein coupled bile acid receptor, also known as TGR5. These modulate hepatic bile acid synthesis, glucose metabolism, lipid metabolism, and energy utilization from the diet. Conversely, gut products such as host and or microbial metabolites and microbial-associated molecular patterns translocate to the liver via the portal vein and influence liver functions. Additionally, systemic circulation extends the gut-liver axis by transporting liver metabolites from dietary, endogenous, or xenobiotic substances, for example, free fatty acids, choline metabolites, or even ethanol metabolites, to the intestine through the capillary system. Owing to this medium of transport and ease of diffusion of systemic mediators across blood capillaries, these factors could affect the intestinal barrier positively for example, with butyrate, or negatively, for example, with acetaldehyde. So basically, this is showing that there is this bidirectional communication and passage of molecules from the gut to the liver and back to the gut. Here we can see certain associated diseases with intestinal dysbiosis. We can see that non-alcoholic fatty liver disease is associated with dysbiosis. We could even look into liver cirrhosis, hepatocellular carcinomas, and see that there is a strong association with intestinal dysbiosis. Now, when looking into the gut-lung axis, dysbiosis in the gut microbiota has been implicated in several lung diseases, including allergy, asthma, and cystic fibrosis. The bidirectional crosstalk between the gut and the lung, termed as the gut-lung axis, is best exemplified by intestinal disturbances observed in lung disease. Some of the existing probiotics, in fact, show beneficial effects on lung health. So here we can see that we have things such as short-chain fatty acids, and short-chain fatty acids can be produced endogenously from our bacteria, and these can actually have beneficial effects not only on the gut, but also the lungs and also the brain. And in fact, you're soon going to find out things such as butyrate has a beneficial impact on the microbiome in every area of our body, but it's only produced within the microbiome of our gut, which is pretty interesting. Metabolites like short-chain fatty acids produced by gut bacteria move through the bloodstream to stimulate immune response in lung, and different factors from lung affect gut immune responses. Apart from metabolites, the immune cells induced by multitude of antigens move through the lymphatic duct between both of these organs, which can lead to the modulation of immune response in both the lung and also the gut. So when there is these short-chain fatty acids produced from the bacteria, they can go into the bloodstream, and that can go to the lung barrier, which can stimulate the immune cells. And then when we look into the lung bacteria, they can produce certain bacterial products, which can then go into the lung lymph nodes, go into the lymph flow, and into the mesenteric lymph nodes, and be passed into the gut. This paper looks at different dietary components and the effects of what they have on the gut bacteria, the metabolic changes by gut bacteria, and the effects on lung health. So when we look into certain things such as saturated fat, we can see that decreased bifidobacteria, and it reduced short-chain fatty acid levels, and it was actually associated with an increased prevalence of asthma. We could look into other aspects such as plant proteins such as whey and pea protein. I'm not quite sure how plant protein is inclusive of whey, but hey, let's go with it. We found that increased lactobacillus and bifidobacteria, which increased short-chain fatty acid, which actually had a beneficial effect on the lungs. It led to this anti-inflammatory response and maintenance of the mucosal barrier. In fact, it even increased T regulatory cells, which therefore regulated the immune system in a more effective manner. So we could pause this here, and I want you to have a little read through this table. There's a lot of valuable information on here. But one thing what I want you to also take into account when you read this is that one man's food be another man's poison. Just because pea protein has been shown to increase lactobacillus, and that could increase short-chain fatty acids, and that could help with lungs, it doesn't mean you should prescribe pea protein for all your clients with issues with their lungs. If they have, for instance, issues with recurrent HSV, well, we know that pea protein is rich in arginine, and arginine can increase viral replication, and that may lead to further outbreaks for them. So in that case, it may not be the best thing for them at that time, at least. Similar to when we look into saturated fats, just because someone has asthma doesn't mean we shouldn't give them saturated fats. If they also have health challenges such as bipolar or epilepsy, we know that a ketogenic diet is going to be preferential. So therefore, we could assist with their asthma elsewhere and still opt for the dietary protocol, which could be inclusive of higher than normal saturated fat intake. When looking into the gut-lung axis further, we can pay attention to this extensive, this extremely detailed diagram between the intestinal lumen through to the lung lumen. And we can see this orchestration of the immune cells and the molecules and how they can transfer and communicate to one and another. Moving on to the gut-thyroid axis, we have to recognize that 20 to 30% of thyroid hormones get activated by gut bacteria. So if someone has subclinical hypothyroidism and they also have dysfunctions in their digestive system, we may not be able to get their thyroid to where we want it to be until we also fix the digestive system. This study looked into Hashimoto's thyroiditis and Graves' disease, amongst other thyroid complications. It found that microbes influence thyroid hormones by regulating iodine uptake, degradation, and enterohepatic cycling. In addition, there is a pronounced influence of minerals on interactions between host and microbiota, particularly selenium, iron, and zinc. In manifest thyroid disorders, the microbiota may affect L-thyroxine uptake and influence the action on the PTU. This diagram gives us a good representation of the interplay between different factors which are associated to thyroid health and also the microbiota. We can see that a disruption to the microbiota can increase the likelihood that obesity will occur. This study is proven in rats. We could also see that the dietary intake could have an interplay on the microbiota, but also the microbiota can have an interplay with the iodine uptake. We also know that sex hormones come into play with the thyroid function, hence why there is 8 times more likelihood for hypothyroidism in females than males because of higher levels of estrogen. And we also know that the microbiota has this interplay with sex hormones. We know that if there is an imbalance in the microbiota, there's a higher chance for the host to have opportunistic organisms. And with opportunistic organisms, there is more likely the case that there is going to be certain enzymes increased. This particular enzyme, which I'm speaking about right now, is beta-glucuronidase. And this beta-glucuronidase enzyme can inhibit glucuronidation, which leads to the enterohepatic recirculation of estrogen, which will therefore compound to higher estrogen levels in the body. On top of that, we know that there are many other mechanisms with the microbiota which can further disrupt the regulation of sex hormones. So, when looking at the thyroid, we have to pay attention to the gut, and when looking at the gut, we also have to pay attention to the thyroid. On top of that, we have the gut hormone axis. Fair enough, the thyroid is a hormone-producing gland, but nonetheless, there are other axes which can come into play here with different hormones. We have things like GLP-1, glucagon-like peptide 1, all the way through to things such as cholecystokinin and even serotonin. And if you pause this here, we have a really nice table which outlines the certain gut activities which can change the state of certain hormones within the gut and the body. We can see from a paper published in 2009 that fasting and postprandial plasma of serotonin concentrations are significantly higher in IBS patients. So we can look into this and question why that could be the case. And it's very interesting when we point this out. We can dive into this in much more detail when we get to the brain health seminar. We could also look into exogenous glucagon-like peptide 1 and how it actually reduces contractions in human colon circular muscle. So therefore there is this direct interplay between certain neurotransmitters, certain hormones, and the gut, which you would expect is how the body works. And we have another table which looks into clinical evidence for gut hormones in functional dyspepsia. And again, we can see things like glucagon-like peptide 1, cholecystokinin presenting with their interplay of the digestive system. Now, here's something that I really want to point out. We have a profile of different gut hormones from methylene, ghrelin, cholecystokinin, GIP, GLP, neuropeptide Y through to serotonin. And we can see the different sites of secretion. For instance, methylene, that's secreted in the duodenum. And this is actually responsible for phase 3 migrating motor complex activity. And it also accelerates gastric contractions. When looking into ghrelin, this is secreted in the duodenum, the stomach, and it can suppress methylene, and it suppresses phase 3 migrating motor complex activity. So as you can see with methylene and ghrelin, there's this delicate dance between one another to orchestrate the movement of the food through the gut. Furthermore, when we move to things such as cholecystokinin, this is again released from the duodenum and it triggers the gallbladder emptying, and it can slow gastric emptying and it can accelerate small intestinal transit. When looking into things such as serotonin, this is released from the whole gastrointestinal tract and it can accelerate gastric emptying. It can accelerate gastric accommodation. It initiates peristaltic reflex and propulsive motility. It can induce slow excitatory postsynaptic potentials and trigger colonic migrating motor complexes. So when we look at serotonin, I'm sure you would have heard the stat that 95% of serotonin is produced in the gut. But a lot of that serotonin is responsible for gut function. As we can see here, it's responsible for gastric emptying. Hence why when we look into the symptoms of people with low serotonin, they are more prone for constipation. And it's why particular products such as Motil Pro can contain 5-hydroxytryptophan, because that can assist with serotonin production to assist with motility to overcome constipation if it was relevant for that individual. So we can see that there is this interplay between hormones, these gut hormones, from one site to the next, responsible for different activities. And if there is a dysfunction with our hormonal axes, there will be a dysfunction with our gastrointestinal axis. Typical dysfunctions or typical causes of dysfunctions for our hormones, we have stress. When there is stress in the body, we really prioritize the HPA axis, the hypothalamus pituitary adrenal axis. And we do so rightly because that axis, that response, that mechanism is responsible for getting us out of danger. It's responsible for keeping us alive. But when we have high stress, we can expect to see a disruption with all of these, what we can see on screen here now. Gut bacteria can influence hypothalamic pathways, having a direct impact on hormone production. So in continuation as what I was just saying with the HPA axis, we have certain bacteria such as E. coli or Clostridia, which can increase epinephrine or they can increase dopamine, and that can further increase that stress-related pathway, which would therefore sway the hormones in another direction. So sometimes we could see that this can become a self-fulfilling prophecy. We can see that the disruption to the gut may lead to an overgrowth of E. coli because there's low stomach acid and there's poor sanitization, and that overgrowth of E. coli has led to increased levels of epinephrine, and those levels are adding to the stress burden, which further disrupts the hormonal production, which therefore further exacerbates the gut function. Moving on, we have the gut bladder axis, and this is why there can be a lot of urinary challenges in some patients which have candida or dysbiosis. It's quite common to see clients experience UTIs when there is high levels of E. coli. In fact, when we look into cranberries, one reason why cranberries work so well is because they contain D-mannose, and D-mannose is this molecule which acts like a sugar molecule, and it enables bacteria to detach from the uterine tract and bind onto the sugar to enable you to urinate it out. We could also see candida is extremely common with UTIs as well, and this just shows this bidirectional communication between the bladder and the gut. Now, hopefully, in an extensive way, this has really pointed out why a keyhole approach to the human body is naive and negligent. Now, I want to reassure you, right now, things may seem complicated. Things may seem challenging, and that's absolutely fine. The human body is complex. We can't become a maestro within 10, 20 minutes, within 2 months, 3 months, 6 months, but it requires patience, understanding, and appreciation. But what I can assure you is by the end of this seminar, you're going to feel much more confident, much more skilled, and become much more valuable for any clients which are challenged with their gut health.